Bismillah ar-Rahman ar-Rahim. We start the fourth video on genetic technology, which is the last chapter of the A2 syllabus. And this is going to be discussed. We've discussed three videos before, and this is the fourth video in which we're going to now discuss the remaining part of the chapter. Now, the heading is genetic technology and medicine. Now, medicine means how are you going to help people with certain diseases? So, the recombinant DNA has been used to, number one, make insulin. So, the insulin is used by whom? It's used by diabetics, people who are diabetes. We have not been able to help in recovery of the pancreas, but all we have done is that we give them injections of insulin. So previously we used to get the insulin from animals. We used to take animal pancreas, cow's pancreas or pig's pancreas, and then of course extract the insulin out of it. But now what we've done is we've genetically engineered bacteria, and now these bacteria are producing insulin for us. So it's a very similar to the human insulin. First of all, we used to use animal insulin. Now that was not very same, and it used to cause allergies in certain people. Plus people used to develop tolerance to it because the body used to recognize it as not its own self, non-self. So what are we using it now? The four main ones which I'm going to discuss today is number one, insulin. Number two, people who have pituitary insufficiency do not make enough growth hormone. So we give them injections of the growth hormone, which is of course given in very early in childhood as soon as it's been diagnosed that he or she has the pituitary malfunctioning. The third is a hormone which is produced for the functioning of the thyroid gland, and that's called the thyroid stimulating hormone. And the fourth is a factor 8. Now these are all proteins, please remember this. So these are all proteins, and we're going to use the recombinant DNA technology. So we're going to get the gene to make these proteins, and then we're going to put it in a plasmid, and then we're going to put that plasmid, which is the recombinant DNA, into a bacteria, and then the bacteria is going to make this protein for us. So please understand the basic biology behind it. Recombinant DNA, and factor 8 again a blood clotting protein. So we're going to talk of four proteins which we can make by recombinant DNA technology. Number one, insulin. Number two, human growth hormone. Number three, thyroid stimulating hormone. And number four, factor 8. Now, which organisms can we use? What is the possibility? Which organisms can we use? Number one, bacteria. Well, yes, a unicellular. So again, it's easy to manipulate it and to grow it also. It doesn't need a lot of space. So number one, bacteria. Number two, yeast. Yeast is again unicellular. Now please remember that bacteria is a prokaryote and yeast is a eukaryote. That is a difference. You must remember this. You must revise what is a prokaryote and a eukaryote. And number three, we can use cultures of mammalian cells. And these of course can be grown in a culture medium, and we can ask them to make, or we can put some genetic genes in it, and they can make a certain protein for us. Now the question is, why use these? Now that's a very simple thing. Why use these? The why is very easy. Number one, they have simple nutritional needs. They do not need chicken biryani every day. They have very simple nutritional needs and large volumes of the product. The product means the protein that they are making. So the large volumes of the product can be produced. But all you have to do is culture the bacteria in a place as big as a room, in a small room. You have shelves and you have the bacteria growing on petri dishes, and they keep on producing that protein for you. So large volumes of the product can be produced. Then you don't need a very big place. It's not a poultry farm or a sheep farm or a goat farm or a cow farm where you need acres and acres of land. You just need one small room, and that's good enough. So not much space needed. Then the process can be carried out anywhere in the world. You can have it on the North Pole, you can have it in the tropics, you can have it anywhere in the world. All you need is a room for it, a building for it. And the fifth is very few practical and ethical problems. You see, whenever in the olden days when we used to use pig insulin, well, we had a lot of religious issues. The Muslim faith did not allow any pig products to be used. So people had issues. Then people who are vegetarians would not use any animal products. So there weren't any ethical issues. And practical problem means in actually the whole setting up of the whole practical, it was very, very easy. So these were the reasons why we used these bacteria and as we just said, bacteria, yeast cell and cultures of mammalian cells. Now, what are the disadvantages? It's a very interesting thing is that bacteria is a prokaryote, so it does not have a Golgi apparatus. Now, if you remember from the first chapter, Golgi apparatus, what does it do? It adds the glycogen part. It is used for modification. So if I have to make an antibody is a glycoprotein. And glycoprotein means first the protein will be made on the ribosome. Then it will go to the Golgi. The Golgi will modify it and add the glycogen part, and then again it will bud off in the Golgi vesicle, and then it will be thrown out of the cell by exocytosis. Now there are some proteins which are glycoproteins. So I'm sorry, then the bacteria cannot be used. Bacteria cannot be used by bacteria as a prokaryote. It does not have a rough endoplasmic reticulum. There is no RER in bacteria. There is no Golgi in bacteria. So these are the ones, this is the one which we need for modification and adding the carbohydrate part or the glycogen part. So then we have one alternative. Let's use a eukaryotic cell. Now the only eukaryotic cell which we have in mind is a yeast cell. Or of course the ones that we said, the mammalian culture, mammalian cells which we culture. So this is the advantage that bacteria cannot. Remember, this question has to come. Why bacteria would not be a very good reason for using recombinant DNA technology to get this protein? Because bacteria is a prokaryote. It has no rough endoplasmic reticulum. It has ribosomes, but those are free ribosomes. It does not have a rough endoplasmic reticulum and it does not have a Golgi apparatus. So there can be no modification, cannot modify proteins. So that is why we cannot use bacteria. We have to use, if you're going to have a glycoprotein, we will have to use a eukaryote. Now what we've done is we have genetically modified. GM means genetically modified. We have genetically modified hamster cells and they produce factor 8 for us. And why do we need factor 8? Because hemophiliacs they cannot produce factor 8, so we need to give them injections of factor 8. So hemophiliacs need it, that is why we are using that. Then in the past what was done is that factor 8 was obtained from donated blood, and this donated blood carried the risk of infection. You could be giving people HIV. So this is the reason why now we have shifted to genetically modified hamster cells and they produce factor 8, and then we're going to give people suffering from hemophilia will be given these. So this was not a very good idea in the past. We used to be getting factor 8 from donated blood, and of course we didn't know, and the blood could be containing any virus, hepatitis virus, AIDS virus. Then let's look at what are transgenic animals. A sheep or a goat could be genetically modified and they produce that protein in the milk for us. And what have we done is we've got human antithrombin and we get it from goats. And this of course stops the blood clotting. Remember, thrombin, prothrombin to thrombin. Thrombin results in the formation of fibrinogen to fibrin, and that helps in the blood to clot. So one of the examples is this. The second example is this. We can also produce human alpha antitrypsin, and we get this from sheep, and it is used in treating people who suffer from emphysema. So these are the two which we have obtained, these two proteins from transgenic animals. Now, what is the definition of a transgenic animal? It refers to an animal which there has been a deliberate modification of the genome. A deliberate modification of the genome. And the foreign DNA is introduced into the animal. And because it is transmitted through the germ line means the gametes, so including germ cells of the animal contain the same modified genetic material. Now, why are we doing this to sheep and goats? Is because that express the foreign proteins in their milk. So transgenic chickens have been produced to synthesize human proteins in the white of their egg. So please understand, what are transgenic animals? Now I would like to talk a little about Dolly the sheep and how she was cloned. So what did we have? We had first a donor cell taken from a sheep's udder. And then what did we have? We had an egg cell taken from an adult female sheep, and then we fused the two. But what did we do is we removed the nucleus of the egg cell. So the egg cell did not have any nucleus. So the egg cell nucleus was missing. Now what have we done? We have put in another cell's nucleus. So the nucleus from this cell is now inside this egg cell. And as soon as this egg cell starts to then fuse, cell begins dividing normally, and then we get a ball of cells. And then this ball of this embryo is placed in the uterus of a foster mother. And this embryo develops normally into, so this is the lamb. Dolly was cloned in this way. So the embryo develops normally into a lamb, and that lamb was the original Dolly, but she was aged because she had the degenerative changes in her. So this is how Dolly, the first clone, was how we managed to get the first animal that was cloned. Now the next topic to be discussed is genetic screening. Now in genetic screening, what do we do? First of all, you've got to understand is that genetic screening would mean something that we would check the person's DNA. Why? Because we want to find out if he or she has a particular allele. Now if you remember from your inherited chain chapter, that people with, if you look at people with different blood groups, the three alleles are IA, IB and IO. Now if I'm blood group O, I would have IO. If I'm blood group AB, I would have IA and IB. So we have different, everybody has the gene for blood group, but the alleles are different. So if I want to find out if this person has a particular allele which can cause a disease, and then maybe I could help him get the treatment for it or prevent it going into the next generation. So who do we genetically screen? Number one, we screen adults. We do screen them for, do they have that allele which can cause the disease? Next, we also screen fetus or embryo, and this is in the uterus. And there are two methods by which we do this: amniocentesis and chorionic villus sampling. And the third that we test is embryos when we are doing an IVF procedure. IVF procedure means that you mix in a petri dish, you mix the ovum and the sperms, and you wait for the zygotes to be formed. So when the zygote is formed, then it will develop into an embryo when it divides by mitosis into 2 cells and then 4 cells and then 8 cells. Now if I can somehow check that embryo, that does it have the disease or not, then I will only implant those embryos which have the normal allele, not the allele which is defective. Now of course any allele can be defective. Sometimes diseases are called by a recessive allele. Sometimes they are caused by a dominant allele. But if we know that, then we would ensure that the embryo with the normal allele is put into the uterus, where it will implant and develop into a fetus, and then of course the fetus will be born and will have a baby who is normal, does not have any genetic disorders. Now an example of screening in adults is if there is a family who has a family history of breast cancer and one of the women in that family wants to be screened. So we can test her, and if she has the allele BRCA1 and BRCA2, we have now identified these, and we can do a test to find out if she has these. And if it is present, then of course she has an increased chance of having breast cancer. So even in a young age, she can decide to have her breasts removed surgically, and that is called elective mastectomy. So this helps this adult to make a decision, which is a very critical decision to make, because then she has to live with this decision for the rest of her life. Because in the years to come, what if a treatment comes for this? And she might regret this decision that she's had her breasts removed surgically. So please remember, this is one of the reasons why we discuss ethics of genetic screening, is that we make decisions, but we're not sure whether those decisions would be valid 10 years from now or 20 years from now in the person's lifetime, and they realize, oh my God, I made a mistake. Why did I get that done? So in adults, this is one of the ways that we screen for genetic screening, is one of these examples which I've given you. Now the next topic is PGD, and PGD stands for pre-implantation genetic diagnosis. Now if you look at the procedure of IVF, what we do is we take the ovums and we mix the sperms, and finally we get the zygotes a couple of hours later. And then the zygote divides by mitosis and it reaches the 8-cell stage. When it reaches the 8-cell stage, we remove one of the cells and we analyze the DNA. Now, when we analyze the DNA, we find out whether it has the correct allele or not. Now, if you look at this example which I am giving you, say the disease is caused by a homozygous recessive. So this person is the diseased person. But this person is the carrier and this person is the carrier. So what we do is we look at the DNA, we analyze the DNA, we find, oh, the DNA, the embryo has this DNA. So, well, we are not going to put this embryo into the uterus then. This DNA, no, we are not going to put this. This we are not going to put this. But this is the only one which has the correct allele, because I said the disease is homozygous recessive. So the people who are homozygous dominant do not have the disease and will not pass on the disease to their children. So embryo with the normal allele is chosen, and then we are going to implant it into the uterus. Other embryos will be discarded. Now there is controversy on this as well. These embryos are potential human beings and we're just throwing them in the dustbin. Is that ethically right? Or can we do research on these embryos and do research of stem cell? And who are the property? Whose property are they? Do they belong to the government or the hospital, or do they belong to the person whose ovum and sperms have been used? So finally, what we're going to have with PGD is we're going to avoid having babies with genetic diseases, because what have we done? We have done pre-implantation genetic diagnosis. We have done pre-implantation genetic diagnosis, in which we have done in vitro fertilization. IVF stands for in vitro fertilization. In vitro means glass. So we've done the fertilization in a petri dish. Normally what we do is it happens in the oviduct naturally. The ovum is released from the ovary, the sperms reach the ovum in the oviduct and fertilization takes place. And then of course this embryo moves towards the uterus and implants. That's the natural way. But in this, what we've done is we're doing this all in the lab. We're doing this in a petri dish in glass. We've done this, we've got the zygotes, and the zygotes divide by mitosis, and then in the 8-cell stage we have removed one cell and we've analyzed the DNA. And then as the disease, I've given you an example, if the disease is recessive, then of course we will not use these embryos, the embryo with this will not be used, this will not be used, and this will not be used. So embryo with the normal allele will be chosen for implantation. Now in this situation, we have a problem for the prospective parents. Why? Because they're difficult choices to make. Then if we find that she is, if of course this we've done in later stage, then we've done this testing, then we're going to terminate the pregnancy. Now any pregnancy in which we do an abortion is a risk to the mother dying. There is a small percentage of mortality all over the world that the woman can die when we do it, even if we do it very properly in a hospital, but there's a prospect of her dying in it. And then sometimes women who've had an abortion will never ever be able to conceive, and that's called secondary infertility, in which she develops some problems, either the oviducts get blocked or something else happens, and she is never able to then have children all her life. So these are very difficult choices for prospective parents. Now, ethics of genetic screening. The UK law allows embryos to be chosen, that you could choose an embryo with the correct allele and then we implant it into the uterus. But it does not allow the addition of an allele to either an egg or a sperm or the zygote. But then other countries have different regulations, so people will go to those countries and have those treatments done. But the UK law does not allow embryos. It allows embryos to be chosen, but does not allow any addition of allele. Because you see, if you add an allele to the egg or the sperm or the zygote, then every cell of that human being which is going to develop from that zygote will have that allele, and we don't know what it will do to next generations. So maybe the UK law is strict about it because of that reason. There are two ways by which we can test the unborn child. One is called amniocentesis and the other is called chorionic villus sampling. Now in amniocentesis, in 15 to 16 weeks of pregnancy, we take out a sample of the amniotic fluid. And we do this with the help of ultrasound scanning because we have to push the fetus to one side and we have to insert a needle, and with the help of a syringe we take out a small volume of the amniotic fluid. Now the amniotic fluid contains cells of the fetus. So we can then examine the DNA of that cells of the fetus. And usually it is done for chromosome mutations. Now the other one is called chorionic villus sampling. Now this is done in 10 to 13 weeks of pregnancy, and what we do with the help of an ultrasound, what we do is that in the uterus when it implants, well, some of the cells start to develop into the placenta. Now what we need to do is we need to take a small portion of this from here. We need to take some cells from here, from the placenta, which is actually the placental part is called the chorion. So the chorion is a part of the placenta. So what we do is we take a portion of this cells from the placenta, and then we take a slide and we examine these cells on a slide, and we look at the DNA of that. So if we find there is a genetic abnormality, then we will ask for the termination of pregnancy. Now there are difficult choices to make. So amniocentesis and chorionic villus sampling both carry a risk of a miscarriage. Miscarriage means is that spontaneously the fetus is going to abort, and the contents, the products of conception, or the little pieces which is beginning, the fetus has just started to develop, that's in chorionic, in 10 to 13 weeks is a very small fetus that has developed, and that can abort. And then of course the woman lives in guilt that she had the tests and then she lost the child or whatever. So is a lot of psychological problems with these two tests as well, amniocentesis and chorionic villus sampling, and the risk of miscarriage is very little, but still there is that risk. And some women may not want to take that risk. They're okay with having a child with or without a genetic disorder. So difficult decisions to make. Now let's look at a disease called Huntington's disease, and this is caused by a dominant allele. So another problem with it is that when the symptoms appear, it's around about by the age of 40 or 50, between 40 and 50, the middle age, the symptoms appear. So by this time people have had children, and then there is no cure for this disease. Now if in a family there is somebody who has suffered from Huntington's disease, now the other people in the family might want to know whether they have the dominant allele or not. So should they test for the dominant allele? Well, okay, if they find out they have the dominant allele, then what do we do? There's nothing they can do about it. Now, so sometimes knowing nothing can be done about it is a better idea not to know that you have the dominant allele. But others might think, okay, fine, if I get to know that I have it, then I might be mentally more at peace. Plus, there is another very important factor is that sometimes people have the dominant allele, but we don't know why, but they never develop the symptoms. So this is one of those lucky ones. So the ethics of genetic screening are very, very controversial. Should we test for genetic disorders for which there is no cure? Should we test and find out and live the rest of the life in gloom and doom? Because until you have a cure for it, or until something can be done about it, should people be tested for that? Should they really know? Would it make their lives better, or would it make their lives sad? Because they would know, okay, well, I'm going to develop this disease and there's no treatment for it, and they've seen other family members suffering and then dying of that disease, and dying a very, very prolonged and after a prolonged illness, and then they have died of it. So they've been sick and they've been unable to look after themselves, they've been bedridden. So all those factors make us think about the ethics of genetic screening. Should it be done for diseases in which we have no treatment and no cure? Nothing can be done to change the genome of the person. We cannot help him or her to change their genetics so that they do not develop those diseases at the age of 50 or 60. So ethics of genetic screenings are very, very interesting yet very controversial. And you can get a question on that, really challenging question on that. Okay, so this is the end of this video, and the rest of the chapter will continue in the next video. Thank you. This video covered the topics of genetic technology in medicine, genetic screening, and the ethics of genetic screening.